everybody. Welcome to Night School. My name's Aria.
And I'm Christina.
We are from the Nightlife team at the California Academy of Sciences in San Francisco, and we are streaming to you from home, but usually we have events in person at the Academy. And we've got a really stacked up program today about the Galapagos Islands, so I'll turn it right over to Christina for some intros.
Yeah, and so why are we focusing on the Galapagos Islands tonight? One, because they're incredibly interesting, but two, the Academy actually has a super long history, over a century of history with the Galapagos Islands, starting with this landmark 1905 expedition, which we'll be touching on throughout the evening. So first up, we have Moe Flannery, who is the Night School MVP, definitely leading for most appearances. So I'm sure many of you know exactly who Moe is, but she is the collections manager of Ornithology and Mammalogy, and she's going to talk about the Academy's stored history in the islands and show us some of the specimens collected back in 1905. And then we have Dr. Jaime Chaves, who is an evolutionary biology professor at San Francisco State University, and he uses our collections a lot for his research. So he's going to talk about his work trying to understand the evolution of a vast range of Galapagos birds, and how he uses collections specifically to do that work. And then finally, we have Dr. Matt James, who is the author of the book Collecting Evolution, and he'll discuss the Galapagos Islands' iconic giant tortoises, which were the main reason that the Academy went on that expedition, and the impacts of this particular collection trip. So yes, thank you for being here. We're excited.
Yeah, we're very excited. And one more quick note on the expedition and everything. April is actually the month that the big earthquake happened, I believe. That's correct. And you'll hear a little bit more about that in the presentations too, most likely. But tonight's program is live. So say hi, let us know where you're from in the comments. And if it's your first time watching, if you're a Night School regular, and stay tuned for questions. There'll be like a Q&A session after every presenter. So get your questions in. And now we're going to go actually to a very short intro video from some of our very own scientists introducing us to the Academy's new Islands 2030 initiative. So yeah, we'll just go ahead and get that started.
One second. Sorry, technical difficulties. Aria, you have to unmute it.
In school, I'm Dr. Lauren Esposito.
And I'm Dr. Rayna Bell.
And together we co-direct the California Academy of Sciences' new Island 2030 initiative. Islands are really the vanguards for how all of life on Earth will face what comes next. They're home to the greatest concentration of biodiversity on our planet and to the highest percentage of recent extinctions. They've really borne the brunt of climate change, and they're also one of the best chances we have to learn how to stop, or even in some cases reverse, the biodiversity crisis.
Islands have always been special, and that's because they're relatively simple and closed ecosystems, and they're where we really learned the basic principles of biology and evolution. More recently, scientists have been realizing that islands are also where we can go to learn about conservation, and especially how targeted interventions to restore habitats and to restore species can scale up to protect entire ecosystems, including the people within them. And so the goal of Islands 2030 is to halt biodiversity loss on 5 tropical archipelagos around the world by the year 2030. And as ambitious as that might sound, this is work we actually know how to do. As scientists and curators at the Academy, we've each worked on tropical islands for more than a decade. But as an institution, the Academy has been working on and studying islands for more than a century.
But the way that we do science, and really who does science, has obviously changed a lot since then. And one of the biggest changes is also what makes the Islands 2030 initiative so powerful. We partner. On islands all over the world, incredible people have stepped up to protect native habitat and species, to increase their community's connection to and knowledge about local nature, and to create opportunities for next-gen leaders. The Galapagos, the Lesser Antilles, the Gulf of Guinea, Madagascar, and the Philippines are all sites of long-standing Academy research, with strong existing partnerships and scientific collections that have already had real impact. This initiative is the culmination of our 170-year history as an institution and of what and who we are today. We're getting to work.
And we hope you'll join us.
Okay, and without further ado, here is Moe in the collections.
Hi, everyone. Welcome to the Birds and Mammals collection. If you haven't been here with me before, we have 100,000 birds and 11,000 eggs and nests here in this room that I'm standing in. It extends way down the hall that you can't see right now. But overall, the Academy has over 46 million specimens that make up the research collections. They're all behind the scenes, except in May, we will be opening a new exhibit on the main floor that is featuring some of the gems of our research collections from all departments. But if you want to learn more about the other 45,900,000 specimens that make up the Academy's collections, you can watch some of the previous recorded night schools on the Academy's YouTube channel. But here in Birds and Mammals, my joy is taking care of these specimens. So I'm like a librarian, but instead of loaning books to researchers, I take care of specimens. And some of the specimens that I am the most honored to take care of are our specimens from the Galapagos. And I'm going to share some of those with you today. So as you heard a little bit about the 1905-1906 expedition, but in 1905 and 1906, 8 men went to the Galapagos Islands on the Academy schooner, and they collected and brought back over 78,000 specimens. A lot of those were plants and insects, similar to the entire Academy's 46 million specimens. There are a lot of plants and a lot of insects, and you can probably guess why. There are lots of plants in the world and tons of insect diversity. They brought back the tortoises that you'll hear about later, and they brought back a lot of bird specimens. And so some of our most important specimens are those birds. I'm going to show you on our camera here. So you can't see, maybe you can there, you can kind of see this entire tray. Oops, let me turn this light off. There you go. Now you can actually see a little better. So these are Galapagos finches, and these are a male and a female, male and a female, male and a female of the different species of Galapagos finch. And they all radiated from the same common ancestor way back in time. And this tray just shows you the difference in different finches, the sizes. So this large ground finch, get him in there, you can see he has a huge bill and a fairly large body compared to some of the other birds. And he primarily, the large ground finches, they would primarily eat hard seeds. They can live in drier areas, and they've evolved to feed in those specific areas. Compared to other finches, and you'll hear more about the vampire finch from Jaime, but we have the small tree finch here, and you can see a lot smaller in size, teeny tiny bill, probably eating a lot smaller seeds than the large ground finch and living in a completely different area. And then let's see, here's no, I'm going to pull out the one that's way back here. This is the cactus finch. So if you compare the bill of the cactus finch with the bill of the large ground finch, hold on, let me see if I can do this with one hand. There we go. You guys see that? The cactus finch has a long, skinny bill here that is most likely used to feed on vegetation and insects. So you can really see the difference in these species. So Darwin's finches, or Galapagos finches, are a really great example of diversity and evolution, and especially natural selection. However, and we can switch off the camera for a second. Everyone knows them as Darwin finches, and they think about, when they think about Darwin and they think about natural selection, they think about the finches. But actually, Darwin, when he was on the Galapagos, he didn't really know what the finches were. He wasn't really an ornithologist, but he collected things, and then ornithologists back in England examined them for him. And so the theory of evolution of natural selection that Darwin is so famous for, and sometimes is credited to the finches giving him the idea, was not actually the finches. He did not know what they were. He didn't recognize them. He thought they were some other species. But I am going to show you the species that really was the inspiration. Okay, now we'll go back to the camera. Yep. So these, if I turn this off of me, these are Galapagos mockingbirds. They're species of mockingbirds that were found in the Galapagos. And they really should get the credit for giving Darwin the idea of natural selection. Darwin recognized them as mockingbirds, and he noticed that they were different from the mockingbirds that he knew from the mainland. And then he also noticed, as you can see from these different species found on different islands, that they differ from different islands. Like this one is a lot, these two are smaller and these two are bigger. Their pattern, plumage patterns are very different. And that is really what gave Darwin the idea of natural selection, was these wonderful mockingbirds. You can see their facial patterns are different and their wing patterns are different. And so the mockingbirds gave him the idea, but the finches are also a great example of natural selection. So I brought a few other birds out for you to see today, and I'm going to ask Jaime to come on and join me for them. Let me just see if I can move this tray out of the way, because Dr. Chaves is actually studying these birds with his students in the Galapagos now. Hi, Jaime.
Hello, can you hear me?
I can hear you. You're going to be, let's hope you can see when I show the birds. Let's see. There's the camera. Who would you like to talk about first, the rails or the herons?
Let's go with the rails.
Okay, so I'll get a little closer to the rails. So I just took out an entire tray of Galapagos rails so that you could see what the specimens look like that Dr. Chaves worked with for his research, which he will talk about a little later. Do you talk about destructive sampling in your talk?
Yeah, we do. One thing that will be cool if you can show us the size of the wings compared to the rest of the body.
Can you guys see the wings on this one?
One of the interesting things about rails around the globe, and Galapagos is not the exception, is that a lot of rails lose their ability to fly once they go to islands.
And you can see why they have this teeny tiny short wing.
And
there's another flightless
the...
bird. Sorry, go ahead.
Yep. No, I was just saying, like these birds, once they get to these islands, they lose their ability to fly. That means that they cannot move from those islands, and they're one of the most highly endemic bird groups that we know of.
They're basically stuck there, right? There are some other flightless birds on the islands too, right?
Yes, one that is uniquely found on the Galapagos.
I think we might be putting one of the flightless cormorants in the new exhibit, but I can't remember. So then these three are some very confusing birds to me today when I was pulling them out of the collection. And maybe Jaime can explain why. I can tell you who I pulled.
Yeah, we're going to touch into a little bit about why the confusion with these different looking birds. And as you can see on the tray there, and hopefully later on in my presentation, they are considered the same species, or even the darkest form up there is considered a subspecies. So what do you think?
I don't know.
Are they
really
different enough to be considered different species? And most importantly, why do you have that dark bird found only on the Galapagos and where the other ones are found? So we'll talk about that.
And just for your information, everyone, let's see if I can read the tag. This one here is from the Galapagos. This one is from the Galapagos, the dark one. And then this one here is from Cocos Island, which is sometimes considered to have the closest relatives to birds on the Galapagos. Was there anyone you wanted to look at in the finch tray? Is there a species?
Yeah, I mean, there's one particular bird in that tray that I am interested in, that is the vampire finch.
Let me see. I pulled those out too.
And this is an interesting group because they were recently described as a different species, because their beaks are very similar on birds from other islands, but they do very different things. They feed on very different items. So there's one of those examples of convergent evolution where you have different species that look the same. And this is one of those groups in one of those new species described as the vampire finch.
Well, we'll learn more about that in your talk. So one other thing people might ask about when they ask about the collection, that was a very fancy tray of finches. Let me show you what a typical tray of finches looks like. Sorry, not that one. This tray. So this is what a typical tray of birds at the Academy looks like. So when Dr. Chaves comes in to work with his students, they will look through all of these birds, individuals, and they'll record measurements, they'll look at plumage, and in some cases, they'll get to take tiny pieces of tissue from the feet. Oh, I don't think I'm gonna be able to show this, but let me see if you can tell. You see the foot of this finch, it has a big lump on it. And so does this one. It has a huge lump right here. It's a little hard to tell, sorry about that. We need a macro lens on this camera. But these two birds have what's called avian pox. And one of the coolest studies that took place in our collection was Dr. Patty Parker came here in 2004. She looked through all the skins because she was studying live birds on the Galapagos and she was studying avian pox. And because the Galapagos is such a unique island and it's always interesting to learn how creatures get to islands, she wanted to know how avian pox got to the Galapagos. So she looked at every bird and wrote down who had pox and who didn't have pox. And then we let her take a teeny tiny sample of those avian pox lesions. And instead of looking at the bird DNA, which is what Dr. Chaves looked at, they were looking at the virus DNA. And she was able to look at the DNA of the avian pox virus from the specimens that had it. And she determined that the avian pox arrived on the Galapagos sometime around 1898, and it was similar to a strain seen in caged canaries. So the story that you could hypothesize, right? We will never really know truly what happened, but based on the genetic evidence, potentially someone traveled to the Galapagos with a caged canary with avian pox, landed on Chatham Island, and then it spread to the other islands according to the genetic signal. So in 1905 and 1906, when the 8 men were in the Galapagos collecting all of these specimens, they were collecting them just for future research. They did not have any idea what scientists would do with the birds in the future. And that's why, for me, the most important thing is to make sure that all of these birds live on into the future. Because in 1906, they never would have believed a female scientist would be looking at the bump on the foot of the bird and amplifying DNA, which they didn't even know about, of a virus in that bump. So we don't know what people will do in the future, but these guys are going to be around to do that. And with that, I think I can answer some questions.
Thank you. And thank you, Jaime, for your cameo appearance. We'll say goodbye for a few minutes. Thank you, Mo.
Thank you.
Fascinating as always. So how do you ensure that the birds specifically in the collection are around for another hundreds plus years?
Yeah, 500 years, 1,000 years, hopefully. Well, this, yeah, exactly. This room is climate controlled. That's why I'm wearing a jacket, because even though it's 80 degrees outside right now, it is 65 degrees in here. The specimens are also in these sealed cases you can see behind me. So they're in the dark all the time to protect them from light. We protect them from high heat, so 65 degrees. And then we try to keep the relative humidity around 50% in this room, which is kind of tough in San Francisco, but those are the goals, to keep them in the correct environmental conditions. We're always worried about pests. Mm-hmm. The 1905 and 1906 specimens were probably prepared with arsenic. They were definitely prepared with arsenic. The tortoises too. That's why I'm wearing gloves, because arsenic is a carcinogen. There's not enough to make anyone sick, but we just don't want to handle them and then eat a snack or anything like that. And these Galapagos birds, I forgot to mention that here, because of that 1905 and 1906 expedition primarily, the Academy and this room houses 60% of all of the Galapagos birds that are in museum collections, at least in North America, that we could survey on a searchable database, GBIF database. So it's particularly important to make sure that this room, you know, is safe in an earthquake and has good fire protection too as well. So,
and outside of the Academy holding 60% of Galapagos bird specimens, can you also talk about why, like, the Galapagos expedition was specifically important to the Academy?
Did I forget to talk about
that?
So while those 8 men were in the Galapagos, the earthquake on April 18th, 1906 happened here in San Francisco, and the Academy at the time was down on Market Street. The building itself survived the earthquake. However, as many of you know who've studied the earthquake, the fires that came through after the earthquake were really what decimated a lot of San Francisco. And so everything that was in the Academy's collections prior to 1906 burned in the fire, even the log books. So we don't even know what we had prior to 1906 because of that fire. We actually have 2 birds in our extinct case that were saved from the fire by one of the botanists, but those are the only 2. So those are number 1 and 2 in our new catalog. And then the rest, the next numbers, the next thousands of numbers, are all the birds that came back with Rollo Beck and his team from the Galapagos. I believe that they got a telegraph that basically said, don't worry, everyone's okay, but you are the California Academy of Sciences. And they really were. The birds they brought back and the tortoises and all the other specimens, the 78,000 specimens they brought back, gave us maybe the courage to continue to grow. And they actually were in the Academy schooner in San Francisco Bay for quite some time.
Mm-hmm.
Matt can probably fill us in on that. I think it was 2 years maybe before they actually came on land, but they helped us rebuild. Why they're so important. And
yeah, Matt has a lot more on that coming up later in the program, but yeah, it's very important. So are there special techniques to using these birds for research, like so that like how do you determine like what kind of samples can be taken from them, but they're still preserved as like these good research grade specimens?
Right. So historically, or in the past, bird specimens were primarily only used for measuring the bills, measuring the legs, measuring the wings, looking at plumage. But since technologies have advanced, like taking genetics from the toe pads, you can look at stable isotopes in the feathers of birds, and you can see where they were living when they grew those feathers. As technology has advanced, we have to be careful about what we loan out to researchers. So we have a destructive sampling committee, and every request to take those samples are reviewed by that committee before they're allowed. You know, and we don't allow sampling from every individual of a species. We always make sure that there's still a lot of specimens that have, you know, fully intact toes. No one ever cuts off much more than a teeny tiny little sliver of toe pad. And they have to have specific laboratories that can process ancient DNA. Similar to like if you're working with old bones, you need a special lab for doing that. The same thing with skin. We're very selective.
Yeah.
But we want the specimens to be used for research. So if it's a good study and they have the funding and they have the resources and a good question, then we'll usually approve it.
That's super interesting. So finally, a very serious question, which is, what's your favorite Galapagos bird that you take care of?
My favorite Galapagos bird, it has to be the mockingbirds. I mean, come on, like they do not get the credit that they deserve.
Somebody said that in the comments. They were like, thank you, Mo.
Yes, absolutely. They totally deserve more credit. Like, forget about Darwin's finches. They're very cool, and we'll learn more about them. But the mockingbirds are my favorite for sure. And they're so gregarious in the islands themselves. When I was there a couple of years ago, you know, they were nesting near our little lodge that we were staying in, or the field station we were staying in, and it was just amazing to see them. They're really great birds.
Great. Well, thank you so much, Mo, for doing night school yet again. We'll see you again, I'm sure. There's so many parts of the collection you tend to talk about, so we'll see you again.
Okay. Thank you.
Thank you. Okay, and up next we have Dr. Jaime Chaves.
Hello, everyone. I hope you can see me now. You can see my slides. So to me, this is a wonderful opportunity to share a little bit of my research and what my lab and my students have been working in the past few months since we started our lab here in San Francisco. I'm originally from Ecuador, and I have been privileged and lucky to be part of this research on the Galapagos for almost a decade now. And today, what I want to present you is some of the work that we're doing to reveal some unknown secrets of the Galapagos. There's so many things that we know already from the islands, but with our research, we're trying to dig a little more in these more obscure species that not so many people have seen, let alone even studied them. And I'm going to be telling a story about 3 of these birds, the rails, the herons, and the vampire finches that Mo already showed them to us in the collection. So let's move on to our first stop. And our first stop is the Galapagos rail. Some of you already saw is a small, chunky little bird, not super colorful, but it's very skulky and it's very elusive. It is hard to find this little bird. And as I mentioned earlier, this is a species that is flightless or partially flightless, meaning that once the ancestors of this lineage got to the islands, they lost the ability to sustain long-distance flight. Nevertheless, it's a very widespread species within the islands. And one of the things that these rails love is this very wet and humid and dense vegetation, as you can see here on the top right, on the higher parts of the islands. There are found around there used to be 9 islands that were reported in those, but there's only 5 of them that we know for sure they're there. And this species was first discovered by the trip and the voyage of the Beagle. So it is thought that Captain Fitzroy himself is the one who collected the first specimen, although there's still some debate if it was actually Charles Darwin himself who did that. So here you have an image of the Beagle and some of the maps from that expedition. And it's in the UK, John Wood, which is the curator, ornithology curator of a Natural History Museum in the UK, who made this beautiful illustration to broadcast the first description of this bird. So this is the first time this bird came to be seen and known to the world. So one of the things that I was mentioning earlier is that this bird is quite, distributed all over, the archipelago. But one of the things that also happened with all these visitors is that they brought invasive species. And in particular, one vertebrate, and you see here in the photo on the top left, goats. Goats were brought, and we're going to learn a little more about later down when maybe Matt will tell us a little about the history of these expeditions. But these goats were left on the islands to roam free and to graze down to the bear the vegetation, the endemic vegetation of these islands, and have a huge impact on the island. So you can see this landscape here on the top in the middle. There is an enclosure of the native forest where goats were not able to go in there. And you can see how decimated the habitat is outside of that fence. And on the right-hand side is the vegetation after removing all these goats from these islands. And you can see that the vegetation is coming back. So you can imagine any type of endemic animal that depends on a very dense forest, how hard it would have been to survive this encroachment of thousands of goats on these islands. To the point that on the bottom map that you see here, the islands with a question mark are the islands that we think these rails got extinct or extirpated due to this intense grazing of their habitat. So we now know that there's no rails on Cristobal. There might not be any in Floriana and in Genovesa. And the populations in these other islands are still very unknown because it's a hard bird to actually see when you go to the highlands. Luckily for many of the species on the Galapagos, there was a super successful eradication program, and we got rid of most of the goats on these islands. But now we are left with the consequences of these introductions. But some of the efforts that are put now to try to save these populations is maybe we can reintroduce, or we can what is called translocate or rewild some of these islands that are now without rails. And not only rails, we're talking about giant tortoises and snakes and even the mockingbirds in some islands. So how do we do that? How can we better inform any type of decisions to pick which one of these bird populations are better suited to those habitats that are now free of goats? One of the things is that, well, we need to match genetically to what used to be there. But you ask me, how do we know what used to be there if there's no more rails there? Well, this is when we connect to the California Academy of Sciences and the expedition of 1905-1906. those specimens back then represent the individuals that were still alive during the early stages of this destruction of the habitat. In this photo here, we have an example of Rollebeck with many of the specimens that are being used. So what we do now, and with one of my students, Pedro Martinez from Ecuador, is trying to first genetically find out signatures of all the rails on different islands. And then we can go to the collection and, as Mo explained, take a little piece of the skin on the leg, as you can see here on this photo of the rails on the top left, and compare the genetics and match to the population within the islands that we now know where they are found and how do they look like genetically, which can be the best suited population that we can use to move some individuals to these islands, because they know that they're going to do best if they are as close as what they used to be. And just to give you an example here in this bottom right tree, this is a phylogenetic tree. This is how you actually are able to define which populations match better the modern samples that we've collected and the museum samples. And the closest they're going to find in one of these branches, the better the match is going to be, that we can support conservation efforts for the Galapagos. And not just for rails, but for many other species from which we now know where can these species come from, and hopefully go back to restoring these ecosystems to what used to be the most close to the natural setting before humans got there. So that is my first story about the rails, and that's how we're doing this research with specimens from the Academy. The second stop is that weird looking bird that Mo was mentioning, that is hard for us to identify what they are, and this is what we call our lava and striated heron. And as you can see here in this photo, on the left hand side we have the lava heron, and on the right hand side we have the striated heron. And these are two species that you can find them on the Galapagos. And a lot of scientists still currently we haven't come to a consensus to come up with an answer. Are these two or one species? And how does it really, one goes around to test that? But more importantly, why there's difference in plumage to begin with? Why do we care if the bird on the Galapagos has this plumage coloration versus the one on the right? And you probably start getting the sense and the answer about that. It's like, just look at the name of the bird. The lava heron prefers to live in the lava, and the striated, well, maybe has better fitness or survival in other parts of the islands. So the way for us to test, first of all, how different they are genetically, we're going to do the same thing that we are doing with the rails. We need to compare the genetic makeup of the lava heron and the striated heron, and we're going to use the same phylogenetic reconstruction to look at to which extent and how closely those species are from one another, or how far they are, and that could give us at least an idea of the separation of the two species. But to do that, we need to catch these herons. And here we have the help of many scientists, including our own Jack Dumbacher from the California Academy of Sciences. And I had to take this picture because it is just probably the first time that I've seen anyone putting mist nets right on the lava, waiting for these lava herons to fly away. Sometimes you have to just flush them so they can get caught in there. And once you catch the heron, then the processing of the data collection takes place. So one of the things that, if you have the chance to go to the Galapagos, and we talk about being this iconic place to understand natural selection, this group of birds is probably, again, an incredible example of how natural selection is operating, keeping these two individual morphotypes or plumages apart. If you walk to the coastlines of the Galapagos, when you see a lot of lava, you might encounter a ton of lava heron with that dark plumage. And the question is, so why these lava heron prefers to go into these rocks And the striated heron usually loves to go into the mangrove. And the answer is right there. They blend perfectly with the background, and they might be better off at attacking and getting a fish or preventing from being eaten by another predator. So you can imagine how natural selection might have operated, where some of the individuals originally got into the island, they might have some variation in plumage coloration. Some of the birds might be a little bit darker than the striated heron. And those birds, once they start wandering on the lava, were doing much better at eating, let's say, than the ones that didn't have that plumage. And by having an advantage versus the other group, those individuals started mating with individuals that were also darker. And their offspring had that information genetically, and they also had a dark plumage, to the point in which every single one of these birds start mating among each other, and the two morphs are now kept in different frequencies on the Galapagos, allowing these plumage to be highly frequent in the coastlines and the striated heron more common on the mangrove area. So if this is not just one of the best examples of natural selection, I know the finches are kind of there, but Galapagos has every single one of these examples, wherever you look around. So one of the things that we also want to do is like, well, can we actually look into the genes that are making these birds to look differently? And in the meantime, I'm just going to put like a little video here of a lava heron, just, like wandering here on the rope, so you can just check it out. But one of the things that we're trying to do, and again, here we have our scientists collecting data for this genetic analysis, is that maybe we can look into the genes that we know are behind coloration differences on other birds. And if you go to the Galapagos, you probably will see the red-footed boobies, and there's two flavors of boobies, the white and the brown morph. And they differ because there's a little mutation in a very well-known gene which is called MC1R. So one mutation in this gene is making that the two populations of red-footed boobies that live in the Galapagos have this different coloration. So this could be another example in which if we look for that gene and we compare the gene between the two species or the two morphotypes, we can come up again with another example on how variation in these genes can lead to this morphotype and this variation that is now kept by natural selection. So, keep your eyes open for any outcome that we come from this research. And the last stop that I have to share with you, and there's again the rail, the heron, has to do with, yes, Darwin finches. And I know that this is kind of the iconic group that we all know when we talk about Galapagos and natural selection and adaptive radiation. But I just wanted to give a little example of what the meaning of adaptive radiation is, and then get into one case study. So the idea here is that you have one common ancestor, a lineage that moved a population of 30 birds that flew from the mainland into these islands around 1 million years ago. And once this population start moving to different islands that have very different ecosystems, these individuals, they had a slight variation on their beaks. They start doing better at eating one item that was more common in one part of the island than in another island. Kind of the same idea with the plumage coloration, in which you have variation and then they start starting out in different morphotypes. The same thing is happening here, but now instead of having different plumage coloration, they're having different beaks. And you can now end up with, in this case, 18 species, all originated from the same common ancestor. And that is a pretty incredible diversity of birds for one island. But one of the most interesting things that I found about is like how different the diets of these birds are. So you have birds that feed on seeds, as the top left ones, like the medium ground finch. You have ones that feed on plants, like the vegetarian finch feeding on some flowers in the top right. Obviously, you have the woodpecker finch that uses a tiny little spike or like a spine from the cactus to retrieve some grubs and some caterpillars. You have the cactus finch that we already saw earlier, long pointy beak to access pollen and nectar, and the weird blood-eating vampire finch. And the research that I wanted to do here is, after reading this quote on this paper, in which the idea that the diet of the birds is something that is correlated with something that we all know very well, which is the bacteria that live in your stomach depends on your diet, right? You have your probiotics, and if you eat something here, you feel a little weird. So there's a tight connection between food and your gut bacteria. So the idea here is that as these species diverge in their feeding behavior, maybe their bacteria also was different, or at least started to differentiate. And so one of the things that we do here is we need to study the bacteria that live inside these birds. So we also go out there and we put mist nets and we catch these little birds. As you can see here, Daniela Vargas catching one of the ground finches. And here I just put a little graph of the steps that we do. But yeah, we need to collect their poop. And by looking to the poop is like going to the doctor. You put your little vial in your sample. The same thing we're doing here. But instead of culturing the bacteria, we're actually doing genetic analysis. So we are identifying the bacteria based on their DNA. And depending on the DNA and seeing how different they are, we can have these, this graph here on the bottom, in which the prediction is going to be that if you plot the genetic differences of all the bacteria, and you say that all the red bacteria are found for the birds that are vampire finches, and all the bacteria that are blue are or belong to the birds that feed on flowers, and the green ones are the bacteria that are found on the birds that feed on insects. The idea is that every single one of these specific beaks have a specific group of bacteria. That is our prediction going into this analysis, knowing how tightly related the bacteria is with the diet of a bird. But when we look into our analysis, the only group of birds that were very different in their gut microbiome were the vampire finches. The other birds have a lot of similarity on the bacteria that they had, but the vampire finches had something outstanding. There was nothing compared of the bacteria type that they live in their guts. And the most interesting thing that we found is that the type of bacteria that is so different is called fusobacteria. So if anything you need to take out of this is going to be this fusobacteria. Remember this tomorrow and talk, in your breakfast, fusobacteria. This bacteria is found and commonly found on vultures and on hawks and on carrion and carnivorous birds. So this is an adaptation that is already found in these other birds in Africa, in Europe. And how on earth this tiny little bird in the middle of the Pacific ended up with fusobacteria in their gut that allow them to do what they do, which is feed on blood. And the birds that are found on the Galapagos, they only live on Wolf and Darwin. I have here my t-shirt just to give it a little perspective. And you have a photo here of Wolf Island. And what they do is pretty remarkable. These are the only bird in the planet known to inflict a wound on another animal. This is not the only bird in the world that feeds on blood. We know the oxpecker in Africa, those birds hanging out on the neck of the giraffes, they're actually waiting for insects to make the little wound or lay eggs so the skin open up, and then the birds will feed on. These birds here, they go right into the wing of these Nazca boobies, as you can see here, and they start pecking at the base of the feathers, which is a very soft tissue, and they start getting all the blood out there. And you can see, the Nazca booby knows it. It's not that he's not, oblivious about that. And you can see like a gang of little vampire finches there waiting their turn to jump and hop on the back of the Nazca booby and start getting their turn for blood. So our studies here is trying to just give you a sense of the unique adaptations and the hidden observations that we can make by using genetics, but nothing like being in the field and connecting with these birds and with scientists. It is most rewarding than this. And I also just wanted to let you know that since we are talking about vampires, and I just want to quote Dracula here, that all these mysteries that we are now solving, we can only get them with more time and with more help and collaboration. So I want to highlight that this work is, a process and years of collaboration with all these institutions And in particular, the importance of museums and natural history collection museums around the globe that provide this type of information that allows us to look into the past, to work in the present, and predict and make decisions in the future. And I also want to give a shout out to my parents in Ecuador, hola papa, hola mama, which, are probably the most influential person in my life. It was through, they taking me to the Galapagos when I was 6-year-old that probably made me being a scientist and being in front of you, talking about my research. So thank you for that. And I want to thank you for listening to a little bit of what we do in our lab. Thank you.
Hi, Jaime. This was great. I know you said remember Fusobacteria. I'm gonna remember the finches hopping on onto that bird. Like that is gonna be in my brain forever. That was amazing. Yeah. But anyways, so the first question that I have for you is, it seems like you've done a lot of work with defining species, or not defining species, but understanding evolution of these birds, obviously. And can you tell us a little bit more about what goes into defining a species boundary when you're looking at these birds?
Yeah, I mean, if you have taken in biology or evolution, you probably learned so many definitions of a species, right? There's around like 20 definitions of species. And if you ask somebody that works with bacteria and someone that studies sea stars and somebody that works with birds, we might probably have a very different approach of what we call species. But the one that I like to use in terms of what the information that I can provide is using genetics and looking at this relationship between the individuals and the populations. So if you have species that are very similar genetically, most likely you will have a very shallow time since the two lineages have split. The farther away they are in the phylogenetic tree, the more isolation mechanisms could have allowed these two populations to diverge. So if they come back into secondary contact, maybe they don't recognize each other. So genetic differences is a metric or a proxy for us to get a sense of how different two populations or two individuals are. And we can probably start drawing the line. But that genetic data can only work if we also add on top of that additional data like morphology, all the data that we see in the museums, the plumage coloration on birds. And something that is very important is their song. That's the way a lot of the birds recognize each other. So if you have birds that are looking very similar, but they have very different songs, it doesn't matter. The female is not going to go for someone that looks the same. He's going to prefer the male that has a song that matches her preference. So it is a combination of tools, but certainly genetics is one of the most powerful that is now elucidating a lot of these species that in the past we thought there were some different or they were not different. Now you look at genetics like 5 years ago based on the genetic data. So it is very cool to revisit these definitions with new tools, and that's that's when museums are so important, and it's just an adventure every time that you want to answer these questions.
Yeah. Oh, I love that. Yeah, that's so cool. It makes so much sense. And so when you're doing restoration work, then can you tell us also a little bit more about the importance of bringing, like in the case of the rails, like bringing similar genetic related birds in the restoration in the context of ecology? Yeah.
Well, I want to say that I am not really involved in any of the restoration programs that are being in place. What I think we can provide as scientists is the information to suggest what would be the best population or the tools to inform those translocation programs. So you can imagine if you have a genetic population that is very similar to an island from which you don't have those anymore, most likely those species are going to do better if you bring something that it was very similar that used to be there, because through years of adaptation, they have probably developed ways to forage on the ecosystem in a way that another population in another island might not have that. They might even have some sort of resistance to different pathogens that other islands in which you still have those populations might have never experienced that. So the closest the match you get, the more chances you have for success. And it's have to do with a little bit of the, bringing someone that already knows the terrain, that has already seen things, right? So you want to bring someone that, making like an analogy, someone that has learned what is to live on these islands, and that information comes from the genetic data. So the closest the match, the higher the success is going to be for the long term and for the success and the establishment of those populations on those islands.
Yeah, that completely makes sense. And one final quick question. So with birds on these islands, are all of the birds that we have documented endemic to the islands, or are there birds that use the Galapagos, for example, as a stop on their migration, or come in and out in some way?
Yeah, that's a great question. So one of the things that is interesting to understand and to see is that most of the endemic birds are usually the birds that are not very good at flying long distance. So somehow by accident, they got to these islands and they got stuck there. And then you have this endemism mechanism to kickstart. So you can imagine birds that are very good at flying. They will stop by on the islands and they keep going on the route to Peru or to Chile. So those birds most likely are not going to be endemic, but they are one of the most important places for feeding or even for reproduction. So we have the wave albatross that is not endemic to the Galapagos per se, but and I would say like 100% of the nesting population is found on Española Island. That's the only place on the planet where they breed. But then they fly when they're not breeding all the way to the coast of Peru to feed. And they're there for months and just hang out in Peru for a long time. And then they come back all the way to the Galapagos to breed and to lay their eggs and to raise their young. So the Galapagos, you have different levels of endemism. And it has something to do about the terrestrial habits or long distance flyers. But whatever whatever bird you're talking about, the Galapagos has certainly an important part in their life cycles. Either a stopover, a reproductive site, or a full-on endemic bird that depends on one particular plant.
Mm-hmm. Yeah, that's amazing. Well, thank you so much for your talk, Jaime. This is amazing. And I'm actually going to bring up Christina again to transition to our next speaker. But first.
Yeah, so next we have some storytelling with Dr. Matt James, who is the professor of geology at Sonoma State University. And he's going to talk a lot more about the history of our Galapagos specimens, since he literally wrote a book on them. But first, we have some footage that's too good to not share with you. It's just a minute. But it's about a couple tortoise-related projects that are happening at the Academy right now. So the first thing you're going to see is our preparator, Ian, doing some careful detail work on a Pinzon Island giant tortoise for our upcoming exhibit, Hidden Wonders. So this specific tortoise specimen is now over 100 years old, and a lot of the leather has started to crack along its face. So Ian's carefully fixing those cracks to make it look more handsome for the public and closer to how it looked in real life.
And now you are seeing our herpetology collections manager, Lauren Scheinberg, using some cool new technology to scan a tortoise. And the 3D models are going to be available to anybody who wants to download them, and researchers can use them to make measurements and do comparative studies with them. And Lauren's team is doing this for a lot of the iconic specimens from the Galapagos, not just the tortoises, which is pretty cool.
Yes, and Moe is also a lead on that project, and so she's going to be helping to scan a lot of the birds that she talked about today. So, okay, and now, last but not least, Dr. Matt James.
Hello, everyone. I'm Matt James, and I'm coming to you from Beaufort, North Carolina. So you guys are all on Pacific time, but I'm on Eastern time. So the story of the Galapagos Islands and the California Academy of Sciences is one that I told in this book. And the work that I do, the writing that I do, dovetails very nicely with what Moe does in the collections, what Jaime does in the collections and in the field, and which work that many other people do in the Galapagos Islands. And so I call this collecting evolution because they were collecting but not actually interpreting anything. But Moe very nicely emphasized how important the collections are now, 50 years from now, 500 years from now, and that's really a very relevant point. So there is a crucial role for the California Academy of Sciences in Galapagos science and conservation. So the science would be things like figuring out which birds are related, figuring out what the names of the birds are or the names of the tortoises. And then conservation, of course, is understanding how we might save things that have become endangered. So the Academy has been involved in this in the past, certainly in the present, and will be in the future. And to me, that adds up to I'm trying to change my slide here, but I don't see it changing. Adds up to relevance. There it is. And so I think that that's a very important way to think of the Academy in relation to the Galapagos Islands. And the material that I was going to talk about the most is the giant tortoises, although this expedition involved collecting plants and snails and a number of other things. And so I include this figure because it is important to show that of the 78,000 specimens that Moe Flannery, mentioned, and a breakdown of who was in what category. So there were shells, and there were animals that were fossils, insects, plants, reptiles, birds, mammals, and eggs that were collected. And eggs and the birds and the mammals are all in Moe's department. But what's important, I think, to note is how few reptiles and birds were collected. So we think of them as being very iconic. They are, but the vast majority of what was collected was shells and fossils, insects, and plants. So in terms of keeping those numbers, in a relative context along the way. So the tortoises themselves were genuinely under threats. The islands back then in 1905 and into 1906 existed without a national park, without any measure of protection. There was no Charles Darwin research station. There were no park guides. There was no tourism. So there was no set of eyes traveling around the islands all the time, keeping track of what might be going on. So the threats were genuine, and the Cal Academy was endeavoring at that moment not to necessarily save them, but to aid in efforts to understand them scientifically. And so tortoises were kept in places like this to sell to passing ships. And so that is an example, a very concrete example of the way that the tortoises were under threat. So the Academy acquired this very beautiful schooner. It was about 89 feet long. It's the same length as Charles Darwin's vessel, the Beagle, although Darwin's vessel was a square rig ship. This is a fore and aft rig ship in the maritime world. And they sought to go to the Galapagos, and they did. They went for 17 months, and they were in the islands for a year and a day. And basically that entire time they were collecting. And the collecting was done by 8 young sailor scientists and a couple of crew members. So there were 11 on board altogether, but the 8 young men, as they come to be known, did the majority of the collecting. And what they did is what is in the collection that Moe studies and Moe keeps track of. And also what they did is the basis for some of the work that Jaime Chaves does. So the relevance is extremely important. We heard a bunch tonight about Darwin's finches and how they may not have been the organism that helped Darwin to understand evolution, but they are the organism that is arguably iconic along with the tortoises and many others. But it is also interesting that the material collected by these men gave rise to the study of Darwin's finches, because without the collection that Moe is in charge of, there wouldn't have been enough material to understand that each island had its own form. Each island had one with a different beak, things like that. Each island had types that would feed on different foods. So the significance of the work of these men was extremely crucial and pivotal in even the fact that we know of a thing called Darwin's finches. They also collected mockingbirds, and they were there in one way primarily to collect giant tortoises. So we can't overestimate we can't overstate the importance of the Cal Academy's expedition. And Moe mentioned that while they were gone, the Academy was destroyed in the 1906 earthquake, which is coming up very shortly, the anniversary on April 18th. So arguably, the Galapagos allowed the Academy to rise literally and figuratively from the ashes and become rebuilt in Golden Gate Park, where it's located today, versus on Market Street, where it was located originally. So that connection is crucial between the Academy and science, the Academy and conservation, and the Academy and history as well. So there are a number of tortoises that were collected and are kept for scientific study. Some are going on display. But what's really important about this is not how many they collected, but the fact that this is an archive of genetic information to be used for conservation in the future. So it allows us to know not only how the species are distributed from island to island, who's on what island, that kind of a thing. But it also helps us to understand what next steps could be taken. If a certain species used to live on a particular island, that could be a focus of conservation. And so those kinds of pieces of information are priceless, because it's just like having books in a library. If those are the only books you have, then you can't replace them. And if these are the only specimens you have, then you can't replace them. The other is that for specimens like this, but also for the birds, with taking the little clipping of material from those pads, the pads that might have virus in them, all you need is maybe the size of a fingernail clipping in order to have enough tissue to amplify the DNA and then to conduct studies. And so many researchers will come in, they'll sample if they're allowed to, and they take very, very little. But the tortoises are a good example of how you need very little to proceed and do quite an impressive genetic study. So that's a way to think of the collections, whether it's plants or insects, tortoises, birds. There are some mammals that were collected. For example, there are endemic rats that only live in the Galapagos, and those were documented in the collections. So all of these materials can be studied for their genetic properties, and it allows scientists all over the world to understand them better. In addition, for the tortoises, it allows us to understand where did they come from here on the South American mainland, but also how the tortoises exist, which island might have which tortoise along the way. And so there have been some very good and sophisticated studies looking at the different forms of tortoises on the different islands. And this is exactly parallel to the different types of Darwin's finches on the different islands. And so it's really important to realize that the studies are parallel. And likewise, this is done with plants. We don't often hear about the plants of Galapagos, but they tell a wonderful story. And the mockingbirds tell a wonderful story. One of the best stories ever is from the land snails. There's a group that almost nobody would have heard of. The little snails that live up in trees on the different Galapagos Islands tell a story that is arguably a better story than the Darwin's finch story. It's arguably a better story than the Galapagos tortoise story, but it's a very obscure because they're snails, and not that many people, really are into snails. Some people are, I am. And so we have to really appreciate the organisms for the stories they tell us, rather than how spectacular they are, or something like that. So what the Academy collected back then includes those snails, and it includes so many different kinds of organisms. They were careful in the way that people were careful back then, but again, because there's no national park, there are no park rangers, there's no research station, it was a very different kind of an environment where things were being threatened and perhaps in danger of going extinct. But the Academy really came to the rescue, brought this material back to San Francisco. And as Moe told us, about 60% of all the birds from Galapagos that are in museum collections worldwide are held by the Academy. So it increases the significance of the collection by having a large amount all in one place. And the Academy does a really wonderful job of preserving and maintaining those collections for future generations. So these collections, for example, of the tortoises, could also be used to decide who should breed with whom. Which tortoise is related to another. And just as Professor Chaves told us, that we could understand the relationships of things to decide who's closely related and who's distantly related. And so that's one of the stories I tried to tell. I hope I did tell in this book. And it's a little bit like in telling the story of an expedition, it's a little bit like putting an envelope in a mailbox, a blue mailbox on the corner, and then we expect it to appear at its destination sometime in the future. But we really don't know, where does it go? Does it go into a canvas bag and then into a truck and then into a warehouse and then to another truck and then down the road, or maybe into an airplane and across the country and then sorted and put into another bag and into another truck and then delivered, paying a bill or something like that. And so in the book, I endeavored to tell that detailed story of what goes on during an expedition. What did they do on a daily basis? And that, I think, helps to inform us about how scientific expeditions work. Because we often hear that there was an expedition and they found all this stuff. And I wanted to tell that day-to-day behind-the-scenes story, because I think that the behind-the-scenes stories are often some of the most interesting, just fascinating things that went on, running aground, sailing in rough seas, being becalmed and drifting for weeks away from the Galapagos, not able to get back because the boat had no engine. The schooner had no engine. So that was my goal. And I thought that by drilling down into the details, it actually would open up the story and allow a greater depth of understanding. And so with these little baby tortoises, they can be used because, we would like to know who's related. Whether it's birds related, plants related, or tortoises related. And so I would say that this collection from 1905 and from 1906 really does help to advance the conservation work of the California Academy of Sciences. And I know that everyone who works at the Academy, the speakers tonight, and others at all levels in the organization really do thank you for supporting the Academy's conservation work. And so I hope that that's a good but brief overview of what happened for a year and a day in the islands, 17 months during 1905, during 1906. And we can really, in a sense, thank this expedition because of it, the Academy continues to exist today. And so I think that to the extent that people might not know that, I think it's really instructive to hear about it and possibly, if you like, to read about it. So thank you very much. And I know that we are scheduled to wrap up at about 20 minutes after the hour. And so I will turn it back over for questions. Thank you. Hi, Christina. I can't hear you.
Oh, I was muted. Here I am. Okay. So first of all, can you tell us a little bit about those land snails? Because we want to allow them to have their spotlight. What do you find so interesting about them? Yes, Chansey says the snail story, please.
All snails matter, and so that it's really important. But they are a microcosm of evolution, and they are all housed in the geology and invertebrates department at the Academy. And since I got my PhD at Berkeley studying snails, they are very much near and dear to my heart. And the first time I went to the Galapagos 40 years ago in 1982 was to collect fossil snails on a paleontology expedition. So they are diverse in the same way that Darwin's finches are diverse. The finches are a little bit different in the beak, a little bit different in size, different in food, and that is mirrored by the snails. But for snails, we're mostly looking at the shape and size of their shells, right? We're not so much looking at what they eat and things like that. And so if you think about what Professor Chaves said was, you have an ancestor who arrives and then change takes place after that. It's the same story for the snails. These ancestors arrive, and now you have a whole cluster of species of snails on different islands. And it's just an obscure topic, but it's really important. And I think that bringing it to the attention of the public is, I think, a good service for the Academy to do.
Great. We'll have to look up some of those shells and share them later on.
There are thousands and thousands of them. So if you take a little collection of them to put on display, they won't be missed too much.
So can you tell us, like, based on, because obviously, well, you didn't get into too much, but like obviously, like expeditions back in 1905, you mentioned a little bit how they collected. It's a little bit different how scientists collect now. What kind of lessons can we learn about good collecting based on going forward from this expedition? Like what? Yeah.
I think that a kind of a rule of thumb for good collecting today would be only do it if you need to.
Mm-hmm.
And if you do need to do it, can you sample but return the organism back into the field? So Dr. Chaves will collect birds in a mist net that he showed, and they might take a feather or they may take a little bit of blood, and then they're released. So likewise for a plant, you can take some leaves, you can take a few flowers, and you won't kill the plant by just taking a few leaves off and a few flowers off of a plant. So many organisms are sampled for blood and just enough to do the scientific analysis, the genetic analysis. And so I think that would be a rule of thumb, that today scientists don't collect unless they need to. But if you're going to collect a snail, you would probably collect the whole thing, and because it's hard to collect part of a snail.
Yes. And then Arnitha, hope I said that right, in the comments is asking, this might be a silly question, but back then, did they collect dead specimens or did they kill them to bring them back?
They did collect them starting alive and then add them to the collection preserved. And this was partly because there was a mindset back then, and this was my challenge as a writer, is to get into the mindset. It's so different than today, where back then it was viewed that everything, especially the tortoises, were fast disappearing, and that it would be just a kind of a shame if they went extinct and there were no specimens in a museum. And putting them in a museum was the only alternative. There wasn't the alternative of having back then a national park or a preserve, or even Dr. Chaves showed us a fence that was around native forests that goats could not get at the native forest. So without those options, your only option is to bring them back. And you can't keep everything alive because you'd have to have food for everything, and the conditions would have to be just right. So it was a matter of bringing them back, transforming them into a museum specimen, but the museum specimen maintains a lot of the value that it can be used in future studies.
Yeah. It's funny because people are still asking about the snails. But Jaime actually popped in to say that as comparison, there are 16 to 18 Darwin's finches, but there are 69 different land snail species on the Galapagos.
I don't know. It's like maybe there's a thing of like snails rock or something like that. You know, snails rule because they are like the masters of evolution in the Galapagos. So the more that people know, the better. I'm thrilled that people are interested in the snails. Thank you.
Yeah. And then finally, like, how did you get interested enough in this topic to write a whole book about it?
It was because of a series of random events, but encountering just some commonly held misinformation, and then wanting to check on it, and then realizing that there was an entire archive of letters and field notes and photographs and publications at the Academy that no one had touched and that were made available to me 100%. And so just as Jaime had an experience with his parents early in life, I likewise had a parallel experience because I lived in the Hawaiian Islands my entire life, from kindergarten through college. And there are parallel examples of birds and plants that evolved on remote, isolated oceanic islands. And so my connection was very similar. And I just was able to really sort of, even though I'm a paleontologist, I was able to put on my historian hat and delve into all of the aspects. And I feel like one of my goals was to really show what wonderful work the Academy has done for all of these years.
Well, thank you. And thank you so much for staying up late to talk with us. I did drop a link to Matt's book in the comments if you want to check that out. But yeah, thanks again, Matt. So glad to have you.
My pleasure.
Okay. Good night. All right, Aria. Okay, so thank you so much to our guests tonight, Moe and Jaime and Matt. Yeah, this was awesome.
Yeah, this was amazing. And sorry, my mic was muted there. But we here at Night School are coming at you on the first and third Thursdays of every month now. So yeah, keep that in your calendars. We will be back on April 21st with a program about nature in the city. So yeah, it's going to be great. Stay tuned.
Yeah, and just to put another little plug for the 21st, the reason we are doing nature in the city is because the City Nature Challenge is coming up. So download iNaturalist and put that on your calendar too. Yeah. But anyway, subscribe to our YouTube channel so you'll be notified when we come back. The recordings all stay there. So if you have some friends that you think would enjoy learning all this amazing stuff about the Galapagos and the Academy collections, share the link with them. And again, thank you so much for joining us, and we'll see you on the 21st.
See ya.
Good night, everyone.